Hello. So we will start in a few minutes. Let's wait for some time and then we can start with the session. Okay, so hello and welcome everyone to the week 3 live session class on fundamentals of protein chemistry, which is being taught by Professor Shagota Dasgupta of IIT Kharagpur. I am Srijan Sen and I will be taking your week 3 live session class. And in this class I would be summarizing all the informations and that were given to you in the lecture in brief details, and then discuss with you a few questions that will help you in solving your assignments, and when the exam comes these questions are going to be very useful for you people. Okay, so I am sharing my slides with you. So my screen is visible to you all. Can anyone of you please confirm if my screen is visible and if I am clearly audible? Am I audible to you people? And my screen is visible, I suppose. Good. So I think we can start with the lecture. Okay. So in the previous classes, we discussed about the basic structure of an amino acid. Then we talked about proteins and how many different types of structures proteins form. So in today's class, we will discuss something related to proteins, that is the protein structure, the stability, and what happens when the protein denatures, and what are the terminologies and the stuffs related with protein renaturation, protein folding, and the stability of proteins. So if we can recollect from the last lectures that proteins can be classified on, or proteins have different structures, that let us assume that it's not a protein, it's a peptide. So in the primary stage we have a sequence of amino acids or a sequence of residues which we consider as the primary structure or the basic structure of a particular protein, and this carries the entire information about a particular protein. Now these residues, so some of the residues can be involved in the formation of different secondary structures. So there are stretches of amino acids which forms alpha helices, some forms beta sheets, some can be involved into loops and turns. So these structures falls under the category of your local folding or your secondary structures. And when these secondary structures club together, or a combination of these secondary structures gives rise to a tertiary structure, which is a consequence of your long range folding pattern of a protein. And finally, when several tertiary structures can form your, say, homopolymers or your heteropolymers, or they can work even single-handedly like your myoglobin, whereas on the other hand your hemoglobin requires three of its other partners to actually bind to oxygen much more strongly. So these entire group of structures falls under the category of quaternary structures, and on a large scale assemblies we can also group these quaternary structures to form the supramolecular protein structures. So as from starting from the primary structure, we can move to the secondary, tertiary, quaternary structures, and hence we can tell that the protein structure is hierarchical. We have talked about this. Now, we have also talked about how the structure of a protein determines the function and what happens when a protein denatures. So, when a protein denatures, it loses its functionality. That is, it cannot function anymore. So, what are the different functions which proteins have? So, proteins have different functions inside our body, which can be related to your, say, regulation of different translation processes, then your different roles in the signaling pathways, followed by transport. Then if these proteins are enzymes, then they can be involved in different kinds of reactions where they can catalyze the reactions and speed up the reaction. Then these proteins can be involved in your locomotion or the movement, and definitely proteins have a particular structure which entirely collaborates to give it a particular function. Now, what determines these protein structures? So we have the energy parameter, which determines how stable your protein is, and then we can study the kinetics, the dynamics of a protein to understand how stable or what is the pattern of the folding of your particular protein. So there are different methods by which you can actually understand that your protein is in the folded state or in the denatured state, and whether or it is in the semi-denatured or a semi-folded state. So there are different techniques. It can be experimental techniques, as well as there are computational methods which actually give you the information about the structure of a protein. Now, what is this protein folding? So the amino acid sequence of a protein determines the three-dimensional structure of your protein as to the folding pattern of a protein. So how do you know that this protein is going to fold or not? When you are performing an experiment, it's very difficult to say by just looking at the sequence, but just looking at the solution, that whether it will fold or not, whether it is a folded protein or not. So what you do it either experimentally or computationally. Now, experimentally it can be determined in many ways. How do you do it computationally? How does it get that information? So it gets that information from this amino acid sequence. So for example, you know which which amino acids would be involved in alpha helix formation, or which amino acids would be more prevalent in your beta sheet formation. So from there you can actually tell, or computational software predicts the structure of a particular protein. So this is totally based on the prediction. However, there are some exceptions that all proteins can be denatured. Some proteins have multiple conformations, and some proteins get folding help from different molecules, especially your chaperones, which are actually your heat shock proteins, or we call them as HSPs, which actually enhance the protein folding pathway and helps in the folding of a particular protein and retain its function. And the function of a protein, as we have already discussed, that it is determined by its tertiary structure. So can we predict the 3D fold of a protein given its amino acid sequence? Yes, it is definitely possible to predict the structure, as done by different computational softwares like your AlphaFold. There are softwares like AlphaFold, then you have Rosetta. There are different computational methods which can study the amino acid sequence or the primary sequence of a particular protein and tell that this portion of the protein is going to be alpha helical in nature, followed by this portion is going to be in the loop region, and then there is another alpha helix and a beta strand following it. So it is entirely determined by the primary sequence of a particular protein. Now, we have also talked about the Leventhal paradox, where, for example, we have a given protein which is of 100 amino acids, and each amino acid or each peptide bond can be involved in 3 possible conformations. Let us assume that it can have n number of conformations, and it can take up, as also we have discussed about the Ramachandran plot, where we talked about what angles of the different secondary structures can the proteins take up to have that particular conformation. So we talked about that. So let us assume for the time being that there is a protein of 100 amino acids and it can take up 3 possible conformations. So we can tell that there are 3 combinations for a particular peptide bond, and there are 100 amino acids. So 3 to the power 100, which comes out to be in the terms of 10 to the power 47 conformations, and that is a huge number. And it definitely takes time for these conformations to for these amino acids or these bonds to attain such conformation. Now if we even assume the smallest second, that is in say in 10 second it requires to sample all the conformations, then it would have taken 5 into 10 to the power 32 seconds. And if we go on the rigorous calculation, we find that for sampling these 100 amino acid sequenced protein, it would have taken 10 to the power 25 years. But this is not the story, because a protein folds quickly into a stable native conformation, and this entire discussion is termed as the Leventhal paradox. Okay. Now this is an overview of the energies or the bonds which are actually involved in your protein folding. So there are 4 types of your non-covalent, or you can call your weak interactions among the biomolecules in the aqueous environment. So here we are assuming that the protein to be in a solution, or say in aqueous condition where there are a lot of water molecules. So the first thing that comes to our mind whenever the protein is in a water molecule is the formation of the hydrogen bond between the neutral groups as well as between the peptide bonds. So in this manner the neutral groups would be forming your hydrogen bonds, and in this manner they would be involved in your peptide bond, for hydrogen bond with the peptide bonds. Along with the hydrogen bonds, there are the most important interactions, which is the ionic interaction, or you can also call them as the salt bridge formation. So ionic interactions can be of, you say, 2 types. One is the attraction and one can be repulsion. So when the close at close proximity, so you have to understand that ionic interactions has to be in a particular angstrom range, and means beyond this particular angstrom the ionic interactions do not actually play an very important role. So these distance between the 2 atoms actually matter. Now when these say you have 2 nitrogens and those both of them carry a positive charge, so these would be involved in the repulsion, and there would be some kind of disbalance between your protein. Whereas if there is an attraction, say your asparagine side chain is involved with an arginine side chain, then what happens? They get attracted and there is the formation of a salt bridge, which actually enhances the protein to be in a much more folded state. So this is the reason why the different kind of interactions are very important. So previously we have also spoken about the other interactions which are involved in the protein folding pathway. One is your disulfide or the covalent linkage, which has a particular strength of 1 kilocalorie per mole. Then there are hydrophobic interactions which occur between your polar groups. Okay, but you have to keep one thing in mind, that these atoms have a particular range between have a particular distance between which these interactions occur. They cannot occur at very high or very when the 2 atoms are very far apart from each other. That does not occur. Okay, so the other interactions are your disulfide bridge formation, which has a strength of 51 kilocalorie per mole. Then you have say van der Waals interactions, which are your weak interactions, which is 1 kilocalorie per mole. Then hydrophobic interactions of 1 to 2 kilocalorie per mole. The ionic bonds have a good strength of 10 kilocalorie per mole, and your hydrogen bonds have range between 3 to 7 kilocalories per mole. So these are the different bonds or the interactions which are very important. very important for your protein folding. Now you say that, now how do you know whether the protein is a stable protein or a not a stable protein? So for example, if we consider these two proteins, if we have protein A, protein B and protein C, now these are involved in their intermolecular stability. Okay, when two molecules interact very strongly, they are highly stable. When they do not interact very strongly, they are not very stable. So what determines that? But apart from this intermolecular perspective, there is something called intramolecular that actually enhances or helps in the folding of your particular protein. Now if we take the example of this, where we have a protein A and a protein B which interacts very strongly, and how do we know that? So there are different experimental methods which tell us, but how will we know that these are actually very stable? These two proteins are very stable. So for that, there are different kinds of bonds which get formed in between these two particular proteins. So if we see that if there are, say, 100 bonds are forming, and then we have 100 bonds are forming between protein, we can see it from two cases. Okay, so let us assume that the first case, there are 100 bonds that are forming between A and B, and 50 bonds which are forming between A and C. So then we can tell that, okay, so 100 bonds between A and B means more stable, and 100 bonds between A and C, that means it is less stable. 50 bonds between A and C, that means it is less stable. So counting the number of bonds, or having a proper understanding of the strength, so we cannot count the bonds because it is occurring in a molecular level. So what do? We calculate it on the based on the strength. So 100 bonds mean, if we have an average of the what is the bond strength, then we can actually calculate it. Now if there are, in the second case, if we have 50 bonds and 50 bonds, so how do we know? So then also we do it on the basis of the strength. So some have, say, ionic bonds, so we know that disulfide bonds have the highest strength. So among these 50 bonds, if there are 20 disulfide bonds in one and 5 disulfide bond in the other, so on the basis of that we can tell which protein is going to be, or which protein complex is going to be more stable than the other. So all these interactions actually play a very vital role in not only your intermolecular protein binding, but also your intramolecular protein folding pathway. And this actually tells us which protein would be more folded in a harsh condition and would be less folded in, say, a harsh condition. Now this assisted folding of proteins, it says that not all proteins fold spontaneously as they are synthesized in the cells. So folding of many proteins is facilitated by a class of proteins which you can call as the chaperones. So these chaperones actually play a very important role in folding of your particular protein, and this enhance your, because you know that not all proteins can fold on their own and require some kind of triggering agents that will help them to fold it. So this role is played by a commonly known agent, that is chaperone. Now these chaperones can be of different types. One class, the class 1 includes your heat shock proteins or the HSP proteins, and these have different classes, say HSP70, HSP40, DNA-K, DNA-J. So these are all your chaperone class 1 types, and this 70 and 40 is the molecular weight of the chaperone. So if a heat shock protein has a molecular weight of, say, 55 kilodaltons, then it will be termed as HSP 55. So this number actually indicates the molecular weight of that particular heat shock protein. Now the class 2 involves your chaperones, and which are examples like your GROEL, then the GROES, and there are other enzymes which are also involved in your protein folding pathway, that is your protein disulfide isomerase, because this helps in the disulfide bond formation, and the peptide prolyl cis trans isomerase that gives it a better conformation, either because we know that the trans conformation is much more stable than your cis conformation, so it actually helps to change the conformation within the protein molecules by changing the different kinds of the bonds, and this will actually help in formation of the different bonds and enhancing the protein folding mechanism. Now the protein, we assume that the protein is folded. Now we have talked about protein folding. Now we have got a folded protein in our hand. Now what do we need to do? We need to denature it. Why is denaturation of a protein very important? Because we have to move in a different pathway to actually understand how well your protein is folded or how stable your protein is. So what tells us that how stable your protein is? So the lesser time. So assume that the more time it takes for you to go from home to school will tell you how far is the distance between your home and the school. And from the home and the market, so you're going to a school which is far away, and from the market you're okay, so home is the common place, and you're coming back from the market, you're coming back from the school. So the more time, so when you're going from the home to the market and home to school, say assume that you're that a car comes and picks you up and puts you down in the market and the school. So how do you know that which is distance is far? Because as of now you cannot tell, because there is a very minute difference. When you're walking back and coming back to your home, that will actually tell you that okay, so from here it is taking a longer time, so it is far away. It is taking a shorter time, so this is close up. Okay, so and also you can assume that someone ties your eyes and you cannot see anything, and you do not have a count, and then they drop you somewhere and they open that eye. That example you can assume. So from this perspective of proteins denaturation, this tells you actually that the more time it takes to denature a protein, the more folded or more stable your protein is. So there are different denaturation assays, like a thermal denaturation, then chemical denaturation. So these assays actually tell you, give you information about how stable your protein is or how folded your protein is and what conformation your protein is having. Okay, so what does this protein denaturation idea tell you? So the conformation of a native protein is only marginally stable. Now conditions different from those in a cell therefore can cause protein structural changes resulting in the loss of three-dimensional structure needed for the function. So this is, you can call as the denaturation. Now what happens? So loss of three-dimensional function is termed as your denaturation. So how do you do that? So you do that using different kind of chemicals, by the heat generated, by thermal assays, anything. Okay, so what happens in the denatured state? The conformation of a protein becomes completely random and the entropy of the entire protein increases. So there are different chemical agents as well as physical agents which you can apply for the denaturation of a particular protein. Now a classical agent is heat or thermal energy, that which has a complex effects on many weak interactions. And on applying heat, you will see that what happens when you're applying heat, the energy in the bonds increases and they break apart, leading the protein to denature. Mostly this when you apply heat, the hydrogen bonds break, the weak van der Waals interactions break apart. So on heating up, proteins conformation generally remains intact until an abrupt loss of structure occurs over a relatively narrow range of temperature. So what happens that you're applying heat at say 30 degrees, 40 degrees, 50 degrees, and then you see that nothing is occurring. So that means that the heat has not crossed that threshold value. So what is that threshold value that has to be crossed for a protein to actually denature itself? So that threshold value or that threshold temperature is termed as your Tm, or the melting temperature. So what is a melting temperature? You assume that you have 100 molecules of your protein. So 50 molecules denature, 50% of your proteins denature and 50% of your protein remains natural. So that is the temperature at which there is a 50% denaturation of your protein, that you can tell as your Tm. So the abruptness of the loss of structure suggested a cooperative process in which loss of structure in one or more parts of the protein rapidly destabilizes the parts of the protein. Now, if you see from this structure, so this is a lot of information. So what is mostly important over here, that the loss of three-dimensional structure of a protein is termed as denaturation. Denaturation, number 2 point is denaturation can be processed by your different chemical as well as physical agents. From the chemical agents perspective, there are different chemicals which you can apply to break the bonds. From the physical point, the most important is your thermal energy or your heat generated. So when you apply heat, it breaks the weak interactions or the weak bonds, especially your van der Waals interaction, then hydrogen bonds, and the temperature above the temperature at which your protein is 50% natured as well as 50% denatured is termed as your melting temperature. So if we see from here that there are two proteins, one is a ribonuclease A and another one is a apomyoglobin. So what happens? So you see that what I have said, so you have to look at the y-axis, which is percent unfolded, and this is the temperature. So to get an idea about the TM or the melting temperature, you have to see at which temperature your protein is 50% unfolded. So if you see that this is the point of your TM and this is the point of your TM for apomyoglobin, and this one is for ribonuclease A. So if you see that at 50 degrees centigrade, your ribonuclease A is melting and it is 50% unfolded. So the TM for your ribonuclease A is 50, whereas the TM for your apomyoglobin is somewhat greater than 60, say assume 65 degrees. So you can, from here you can get an information. So based on the denaturation step, you can comment on the TM as well as the folding of a particular protein. So the higher the TM value, the more stable your protein is. Okay, and a chemical reagent can be your guanidinium hydrochloride. So guanidinium actually is responsible for unfolding of your protein and bringing it to the soluble fraction. Okay, so sometimes we use it when proteins are very much insoluble in nature and comes to inclusion bodies, we apply guanidinium hydrochloride. So anyway, so for denaturation of a protein, we actually provide guanidinium hydrochloride. So increasing molarity or increasing concentration of guanidinium hydrochloride will actually help in the unfolding of your protein. But anyways, 50% protein unfolded is your TM value. So TM, because here we are applying the heat, but for guanidinium we cannot comment on the TM, but we can comment on the stability of a particular protein. Now proteins can be denatured by extremes in the pH by miscible organic solvents such as your alcohol, acetone. There are solutes such as your urea, guanidinium hydrochloride, which denatures your protein, and also proteins get denatured with different kinds of detergents like your SDS. Okay, sodium dodecyl sulfate. So but what happens is that there are a number of agents which can be used to denature your protein or to open up your protein, but these agents are never involved in breaking of your covalent bonds. Okay, like what are the covalent bonds which can come to your mind when you think of a particular protein? So the first thing that comes to my mind is the peptide bond. Okay, that is formed between your amino acids to give it the primary structure of your protein. The second one that comes to my mind is the commonly found covalent bond is your disulfide bond. So these bonds remain mostly intact because these are not broken by your these denaturing chemicals like your urea or your guanidinium, or you provide some physical agents like your physical agents like your heat, thermal energy. These are not going to actually break up your covalent bonds. So the covalent bonds remain as it is. It only breaks the weak interactions or the weak bonds that are involved between two proteins or intermolecularly. Okay, so urea. So what are the things that we observe? So organic agents like your urea and detergents act primarily by disrupting the hydrophobic interactions that produce the stable core of globular protein. So you can there might not be questions, but you can be asked like what breaks what bonds. So urea and detergents break your hydrophobic interactions. Okay, urea and guanidinium hydrochloride also disrupt your hydrogen bonds. So extremes of pH alter the net charge on the protein, causing electrostatic repulsion and the disruption of the some hydrogen bonding. Okay, so just keep in mind the names of the reagents and which bonds or which interactions do they disrupt. Now the denatured structures resulting from the various treatments are not necessarily the same. So lastly, the denaturation often results in aggregation and precipitation of your unfolded protein, and the protein precipitate that is seen after boiling an egg white is as well a known example. So if you see a real life example, it is the protein precipitate, that the aggregation of the protein that you see after boiling an egg white. So that is also a protein that when you apply heat and boil it at high temperature, it denatures and it aggregates, giving you a solid structure. Okay. Now coming to the renaturation of your protein. Now what does this convey? So first we have denatured the protein. Now there has been different studies where you have seen that there is a native, where you have a native structure of a protein which is catalytically active, and now in the presence of urea, so what does this urea do? It breaks up your, what did we study? So it breaks your hydrophobic interactions as well as your hydrogen bonds. So urea breaks those. Beta mercaptoethanol or the mercaptoethanol is involved in breaking of your covalent bond, especially your disulfide bond. Not all covalent bonds, only your disulfide bond. So what does this beta mercaptoethanol do? It breaks your disulfide bonds, and these sulfur groups remains in the reduced state. Okay, so first we have a catalytically active protein, and on the application of these chemicals, it gets unfolded and inactive, and the disulfide crosslinks are reduced because of this beta mercaptoethanol. And on the removal of urea and mercaptoethanol, you get this active state where these just when you're removing the reducing agent, these disulfide bonds gets, these sulfur atoms gets oxidized, and they form bond in between to give you the disulfide bond. Now what this person Affinson, where did we see that term of Affinson? What did this person actually do? Okay, so there had been studies where what this person did was he actually applied urea, and then it allowed the protein to renature it very fast. Okay, so when the denaturation process was very fast, he observed that random molecules are actually forming your uh, disulfide bond. So it is not giving the protein a very stable structure. Rather, when he removed that urea and the beta mercaptoethanol and allowed it to undergo that protein folding process in a very slow manner, or in a manner in which the protein wants to, meaning leaving it upon the protein to actually undergo that protein folding pathway, then he found out that only those disulfide bonds which gives your protein a much more stable structure, that will be actually involved in your protein folding. Okay, so only those which can form disulfide bonds leading to the proper folding or giving it back to the native structure of a protein, those will be involved when you let the protein to go to that folded state in a much slower manner, rather than forcing it in a faster manner to undergo that protein folding manner. So anyways, when you are doing an experiment, keep this thing in mind that a protein wants to fold it in a manner they want, rather than if you want the protein, you want a result say in just a few hours, and you let the protein fold by providing some harsh chemicals, you know that you give an oxidative agent and you let the formation of these disulfide bonds, but this you will find that the protein may be folded, but protein might form bond, but it will not be properly folded. So you have to follow that slow renaturation pathway for proteins. Now there is a question for you people, and you answer me in the chat box. Okay, so assume there are 8 cysteine, um, there are 8 cysteines in your say protein molecule. Okay, in your protein there are 8 cysteines. So how many possibilities are there for the protein to form these disulfide bonds? I guess my question is clear. There are 8 cysteines in your protein molecule. So in how many possible ways the disulfide bonds can be formed. Okay, so I'm writing the question. Tell me quickly. It's a very easy question. There are 8 cysteine molecules, so how many possible combinations of disulfide bonds will form? Anyone who can tell me the answer? I guess everyone of you are awake. Okay, that takes some time to solve it. I can look out for the answers after we complete the lecture. Okay, never mind. Find the answer, because I'm not going to give the answer of this to you, because it's a very easy question. Just think as to in how many possible combinations these disulfide bonds can occur. Okay, so you will get the answer. Okay, right. So think of it and let me proceed. Okay, so what does this early study have examined? The refolding of an enzyme ribonuclease, which provided the experimental proof that your tertiary structure of a protein is determined by the primary structure. Now, what happened in this experiment was the purified ribonuclease was denatured to its unfolded inactive state by, say, agents of urea and mercaptoethanol. So whatever has been written over here is just has been shown over here is portrayed in the writing part. Okay, so then the unfolded proteins and the reduced disulfide cross linkages comes to the cysteine residues, and when urea and mercaptoethanol were removed, the randomly coiled denatured ribonuclease spontaneously refolded to its catalytically active state. Now even the protein's 4 unique disulfide bonds reformed correctly. Now this experiment provided the first evidence that the amino acid sequence of polypeptide chain contains all informations required to fold the chain into its native three-dimensional structure. We want the native form back. It is not any structure that we are looking for to, that it forms a random disulfide bond and it occurs, but we are looking to the getting back the native state of the protein. Okay, so the native state of the protein is what actually matters. Okay, now coming to the thermodynamics of protein folding. Now what occurs in a, from thermodynamic perspective, if we see that it can be considered as a free energy funnel, where we have the unfolded state on the top of the funnel and they have very high energies, say your enthalpies and the entropies, because the proteins are always in a random motion in your solvent environment, and these are characterized by a high degree of conformational entropy and relatively high free energy. Okay, so now as folding proceeds, so you are wanting the protein to be together. You want all the bonds to be such constrained that they can only rotate about in particular few conformations. So what does this do? You are narrowing down the funnel. You are wanting the energy to be very low. Okay, so you are decreasing the entropy, you are decreasing the enthalpy, you are decreasing the free energy. Okay, so that will actually help the funnel to narrow down in a conformational space that must be searched as the protein approaches its native state. So when the protein is unfolded, it is on the top of the funnel. Okay, it is on the top of the funnel, and then when it starts folding, it actually narrows down because the only few conformations are important for the protein folding, and one folding process actually helps the folding of the other portion of your protein as well. So if you see that here the entropy is high, and as you move down the funnel, so this is of high energy as well. So as you move down the funnel, there is discrete folding intermediates, and you say the percentage of residues in the protein in the native formation. So most of the protein the residues in this protein would be in the native formation in this region, and at the tip of this funnel you get the native structure of your particular protein. So what we see that this is the funnel. Here we have high entropy molecules, and the in free energy would be very high. Then it goes through the molten globule state where it folds, and there is the intermediate state, and then as it moves close to its native structure, then we get the native form, and the funnel actually narrows down. And here you can see a few of those sharp rises and sharp peaks, because you see that a bond is getting formed and then again it detaches, another bond is getting formed, it detaches, because that is not leading to the native structure formation. So because of that breaking, joining, breaking, joining, breaking, joining of unnecessary bonds, okay, that will actually give you a few peaks. So you go down and you say it's okay, then you see no, it's not okay, it goes up, and again the entropy increases, and then in this manner you get this sharp bends. Okay, and at the tip you get the native structure. So I have spoken about chaperones. So many proteins do not fold spontaneously as they are synthesized by the cell. So the folding of these proteins requires general class of proteins called your chaperones. Now these chaperones interact with partially folded or improperly folded polypeptides, facilitating the correct folding pathway or providing you the environment, okay, where these folding process can occur. And you can find these chaperones in several organisms, in eukaryotes, where like your prokaryotes, like your bacteria, to human beings, and these. So the two major classes of chaperones which are actually involved are your HSP70 and your chaperones, your Growell or the Growes, as I was speaking, and your HSP60 family of proteins. So these HSP stands for the heat shock protein, and these proteins are induced in bacterial cells to expose at high temperatures. So at high temperatures they become active, they are activated, and they undergo the protein folding pathway. So molecular chaperones act as monomers, where chaperonins are large multi-subunit complexes. Okay. Now let me see. Did I get an answer? Am I audible? Hello? Am I audible? Good. So how far have you people heard? Because I felt that my mic was off. Did you hear the routine, means the funnel, inverse funnel? Till which slide number did you people actually hear? Can you confirm me? Then I can repeat the other portion. Till this much was audible? Or till this much? Till which state? Means, sorry, till which slide did you actually hear? Can any one of you tell me till which slide? Okay, anyways, I'm repeating the funnel thing another time then. Okay. So, just repeating the funnel portion. So in the top of the funnel you have the state where the proteins would be mostly unfolded and the entropy would be very high. Now as you go down the funnel you pass through the molten globule states and you would be seeing that there are few bends and peaks where you would be seeing in this portion. So these are because of the non-native bond formation, where there are different kinds of bonds which are forming and breaking, and because of that the entropy of the system is rising and again falling to the native state conformation, and because of this you would be seeing a few bends, and in the top you have the protein is unfolded and the entropy as well as your free state would be free energy would be very high, and as you're going down the protein is becoming more and more folded and the entropy is decreasing, and at the end of the funnel you would be observing the protein to have the lowest entropy as well as uh, it would be in your native conformation. So from the bottom to the top the energy is increasing, and from the top to the bottom your percentage of residues of the protein in native conformation is increasing. And we have also talked about the chaperones which are actually involved in the spontaneous formation of your protein folded structure, and these requires and there are different kinds of chaperones, like you have the class 1s where you have the HSP70 and the class 2 where you have the chaperonins. These are the commonly used chaperones, and then you have the GROS and the GROEL of the class 2 molecules. And these proteins are mostly active under high temperature. So we call this as the heat shock protein. So when they are at high temperature, these proteins becomes more active and allow the protein folding pathway to undergo spontaneously. Okay. Okay. So up till this much was your protein folding, protein stability and all. After the 5 minutes of break we would be talking about the protein structure determination, and after the structure determination we can discuss questions. So I have 3 more lecture slides to discuss the lecture, and then we can we can discuss the question answers. Okay, and in this 5 minutes break kindly find the answer to the question which I provided. So there are 8 cysteines which are present in a molecule or a protein molecule, and how many possible combinations of the disulfide bonds you can find. Okay, is my question clear to all of you? Then find the answer and give me a number in the chat box. We will start at 7:05. Okay, so I only got one answer. So 8 cysteines of 4 disulfide bonds possible. That is true. That is true. That is the minimum number. How much maximum? I said what are the possible combinations of disulfide bonds you can get. No one else was able. Okay, so this is a totally a concept of probability. You have done this numericals in your class 11 12 maybe. Okay, so there are 8 disulfide bonds, say 1 2 cysteine 1 2 3 4 5 6 7 to 8. Okay, now one can form with 2, one can form with 3, one can form with 4. So how many possible disulfide bonds can it form? So for the first cysteine there are 7 combinations. Okay, I guess you people can see the chat. So there are 7 combinations. So say after the 7 one has combinations with all the 7. Now cysteine 2 is already bonded to 1. Now when one disulfide bond is formed, so say between 1 and 2 it is formed. Now a disulfide bond cannot form between 1 and 2. Now there are 6 other cysteines left. So between these 6 cysteines there can be 5 different combinations. Yeah, because 5 different types of disulfide bonds can be formed. Now when the second disulfide bond is formed, so 4 disulfide bonds will be formed, but they can form in different ways. So when none of the disulfide bonds are there, there can be 7, because there are 8 cysteines and 7 disulfide bonds. Now when it reduced to 6, because one disulfide bond is already formed, it becomes 5. Okay, now 3, lastly 1. So how much does it add to? So we have 7 into 5 is 35, 105. So there are 105 possible ways in which a disulfide bond can be formed between your 8 cysteine in a protein molecule. I guess it's clear to everyone. There will be 4 disulfide bonds, but the possibility of the disulfide bond formation is 105 ways. Clear to all of you? Okay, so I guess everyone is back. Okay, let me carry on. Now we have to, so we talked about the protein folding, protein denaturation, renaturation, we talked about it. So how are we going to understand what structure this protein molecule is actually having? So for that there are different experimental methods, especially your X-ray crystallography, where you have an X-ray which goes and strikes the electrons, and these have different scattering patterns, and this pattern is repeated based on the plane on which these electrons are present. So you have to keep this in mind, that there is a, for X-ray crystallography, there you get scattering patterns, and this pattern is replicated on your detector or the screen on which you are observing, and this pattern is based, or the X-ray goes and strikes the electrons on a particular plane, and there are different unit lattices, and also that much details you will get when you look out for the lectures. So on a primary basis, on a basic note you have to understand that X-ray crystallography is important for the structure prediction. Then you have the NMR crystallography, where you study the spin states of the nuclei, and this gives a peak based on which you can tell as to what is the status of the protons or the carbon atoms, and your so there are different experiments by which you can perform and comment on the structure of a particular protein. Okay, then there are computational methods of structure prediction, where you have the AlphaFold, the Rosetta, then you have the Holy Grail. Okay, so the sequence implies your structure. Therefore, in principle, we can predict the structure from the sequence alone, because you know what amino acids will fold in what group of amino acids will fold in what manner and form what type of a structure. Now then you have the ab initio method, which uses just the first principles like your energy, then the geometry and the kinematics, which will tell you about the structure of a particular protein, and then the homology pattern, in which you find the best match to the database of a sequence with known 3D structure. So another new structure generation technology is your Rosetta, where new structures are generated. So you have this Rosetta, where you generate new structures by swapping the compatible fragments, and these accepted structures are based on your energy and the structural size. So when you run the particular protein molecule from the PDB file in your Rosetta, so okay, then you get the stability of the structure, and you get this you actually get the free energy value. So the more negative the value is, the more stable the structure is. So you get a lot of clustered structures, so conformations, and based on that you have a list of only those conformationally stable structures, and then you collect the base structure and proceed with your different experiments or what you want to do with that particular structure. Now the best cluster is one with the greatest number of conformations within 4 angstrom deviation structure of the center. So from now when you run a molecular dynamics, you can also get the RMSD and the RMSF plot and all. Now the representative structures taken from each of the best 5 clusters are returned to the user as per prediction. So now when you are running say 100 nanosecond of a particular protein, so this is more of a complex thing. Okay, so you will understand this when you hear a lecture of say computational biology or something. So if in brief I tell you, you put a structure in Rosetta and you run it for say 100 nanoseconds. Okay, sorry, you run 100 structures. Okay, you find 100 structures. So will you get the 100 structures? No, you actually run 500 structures, and based on the best of the 5 you pick up one. Okay, so you run say 100 structures, so actually you are running 500 structures, and from 5 you can pick up one, so you get 100 structures from the base of the 5, and based on that you are predicting the structure or the stability of a protein. Now what is homology based prediction? So it has to show an sequence similarity of something above 30%, and when you superimpose the aligned sequence onto the structural template or the main protein molecule according to the computed structural alignment, you have to at least get a sequence similarity of 30% to actually find the structure based on the homology prediction. So you know a particular protein to be folded in this pattern, a particular pattern where you have say 5 alpha sheets and 5 alpha helices and 2 beta sheets. So you know that it has about 90% similarity with this protein, so it is going to fold in this particular manner. So there are different classes of the same protein which folds in a particular similar manner. So how do you know that? So it is based on the structure or the homology based prediction. So the perform local refinement of the resulting structure in three-dimensionally. Now the number of unique structural folds is small, possibly a few thousands, and 90% of new structures are submitted to PDB, that is the protein database bank, and in the past 3 years there has similar folds in the PDB file. Okay, so this is all about the structure prediction. and in details, when you go through the lectures, you can get informations about these in much details. So I would be moving on to the most important part, where I would be discussing the assignment, sorry, not the assignment, the questions which you can expect in the assignments and the exams. Okay, so the first question, so you people would be answering me, and I expect every one of you to participate in this is not a very hectic job, so try to actually come up with an answer. Never mind if it is wrong, but try to participate in this portion of the class. Okay, so the question one, which says, between which two residues would a salt bridge form? So between which two residues would a salt bridge, or the formation of a salt bridge, is um, more probable? You can write the answers in the chat box. I guess my slide is visible and you people can answer. So I need the participation from all of you. So try answering these questions. So a salt bridge is found between which two amino acids? This is an easy question. Try answering it. Every one of you are there? So a salt bridge is formed formed because of the ionic interactions between, say, a positive charge and a negative charge, as I've spoken. So if you can recollect what are the acidic amino acids and the basic amino acids, and which have a charge in the side chain, so you can recollect that there is glutamic acid, aspartic acid in the acidic category, and you have histidine and arginine, lysine in the basic category. So if you see, in the first place we have glutamine and leucine, then we have aspartic acid, lysine, aspartic acid, arginine, and glutamic acid and lysine. Okay, so the question says, in which of the following residue pairs does not form a salt bridge in proteins? So which does not form a salt bridge? So aspartic acid and lysine, negative positive. Glutamic acid and lysine, negative positive. Aspartic acid, arginine, negative positive. So all of these three would be forming a bond. Okay, salt bridge. But the one that would not form is your glutamic acid and the leucine. So that's why A is the correct answer. Okay, question number 2. Point mutation in hemoglobin causes sickle cell anemia by changing a glutamate residue to a valine. Which type of protein structure is affected by sickle cell anemia following this mutation? So I've spoken about sickle cell anemia in the previous class. Answers in the chat box. You have to try answering this in this live session class. This is an interactive class where you people have to interact with me. Otherwise it becomes that normal lecture which you hear. So these classes are actually organized for you people, so that you people can ask questions and clear your doubts. So you have to be more interactive rather than the person who is teaching you. You have to come up with questions and answers during these classes, and these are interesting questions. This is not much tough. Try answering them. So what kind of abnormalities do you observe in the protein structure because of sickle cell anemia? Or what particular protein structure is getting affected because of this? So if you see that glutamic acid is a negatively charged amino acid and it is getting converted to a polar amino group or a hydrophobic residue. So what type of interactions can it hamper? It will affect the ionic interactions, because of which your secondary structures are actually getting formed. Also your tertiary structures would form because of this hydrophobic interaction, the alpha helices, the beta sheets. Beta sheets are because of the hydrogen bonds, but look at the ionic bonds which is formed in the alpha helix, so that the secondary structure is affected. What about the primary structure? Yes, the primary structure is also getting affected because the glutamate residue is being converted to a valine. So there is a change of amino acid, there is a change of information. So because of that, primary structure is also affected, the secondary structure is affected. So how does the tertiary structure get affected? So ionic bond ionic residue, or say an acidic residue, is converted to a hydrophobic residue. So hydrophobic interactions might increase because of that. So there is a change in the tertiary structure as well. So because of that, 1, 2, 3 both are correct. So there will be change in the primary, secondary and the tertiary structures. Okay, I guess that is clear. Question number 3, which says, which of the following options contain all the information necessary to fold a polypeptide chain into three-dimensional structure? Is it the DNA sequence, is it the histone, the amino acid sequence, or the RNA sequence? So what determines the conformation of a protein? So what are the building blocks of proteins called? So the primary structure of a protein is formed by the amino acids. So that carries all the information of your protein folding. So the amino acid sequence determines the uh, how your protein will fold, or determines the 3D structure of your particular protein. So in X-ray diffraction pattern of proteins, what do you observe? Is it the molecular density map, the electron density map, the nuclear density map, or the atom density map? So if you look into a crystal structure of a protein, you would be seeing that there are different planes based on which the scattering pattern is observed, and this scattering pattern is replicated. Because when you look into a lattice, the basic unit of a lattice is a unit cell. So this unit cell is repeated in patterns, and this unit cell, one information in a unit cell is similar to that in the other unit cell. So there would be repeating patterns. So what are present in this unit cell? So these are the electrons that actually matter. So when you actually diffract, so there is a diffraction pattern because of these electrons present on a particular plane. So because of that, you get different diffraction, different X-ray diffractions, and it can be understood when you look at the electron density map. So the electron density map is a three-dimensional plot that shows you the location of the space electrons within the repeating unit in the crystal, and it is the end result of an X-ray structure determination. So when you get the structure, means you from this map you can find the structure of a protein. Okay, so now this question is from the above mentioned curve, melting temperature of the protein would be. So what would be the melting temperature, or the TM, of the protein from this graph or this curve? What can you comment on the TM? So how much will be the Tm? Good. So I got one answer. You are saying B. Any other answers? So Tm is the state when a protein is 50% folded or 50% unfolded. So if you see at which the protein is folded, so we have 0.0 and then the fractions of the unfolded proteins. So at 50% means at 0.5, how much it is coming? Okay, it is going over here. So it is coming at around 45. So 45 degrees centigrade is the correct answer. Akash, it will be C. Kindly verify. Okay, because when the protein is 50% folded and unfolded, at that point, that is the point of your. So this is the point of your Tm. Okay, so this is the point of 45 degrees centigrade where your protein would be 50% folded as well as unfolded. Yes. Okay. Now coming to the next question. The molten globule state of a protein is one of the states in the folding process. Which of the following option is incorrect about the molten globule state? It is a denatured state. There are hydrophobic interactions among the nonpolar residues. It is less compact. It is a collapsed state. What is a molten globule state? So this molten globule state lies in between the top part of the funnel and the bottom part of the funnel. It lies in the between. So it is not fully unfolded, not fully folded. So it can be considered under the denatured state where there are several bonds which are forming as well as these are breaking. So this molten globule state is, you can call it a partially denatured state where interior side chains are improperly packed and less compact than the native state. So because of that, it is a partially denatured state, not a fully denatured state. Okay. Now collagen is found in connective tissue such as tendons, cartilage. Now collagen provides the strength due to. So if you have studied about collagen, then you can find that. Okay, tell me what determines the strength of the collagen? So if you look into collagen structure, so firstly it has a primary amino acid structure of glycine, then X and Y, where your X and Y is your proline and hydroxyproline or hydroxylysine. So those are repeats of that, and this peptide chain is actually forms a tripeptide or a triple helix. Okay, so these triple helix, so these are alpha helices and these are forms a triple helix. This primary structure forms a triple helix, and then there are different cuts at the terminal ends, after which you get a very compact fibrous material which you call as the collagen fiber. And the strength of this collagen is entirely on this alpha helical, depends on this alpha helices, because there are there is a triple helix that is formed, and this triple helix determines the structure of your collagen, and the fibrous protein is stable. Right, so question number 8, in the zinc finger motif, which residues participate in the coordination of the zinc ion? So which residues can form a coordination complex with your zinc ion in the zinc finger motif? So you can find these, okay, I got an answer, good. So you have to study the lecture because before coming to this interactive sessions, that will actually help you, because these are the questions if you can answer on your own, you would not only help you in solving the assignments and the exams, but also help you to boost your confidence. So try to give an answer. Here no one would tell you anything if you're wrong, but if you're giving an answer, that will also help you to clear your doubts. Now if you do not give me an answer, it would be very difficult to actually understand whether you people are understanding or not, and then the sessions would make no sense. Anyways, so yes, that is the correct answer. Cysteine and histidine are the two residues that act as ligands to the zinc ion, and these zinc ion is important for the stability of the tertiary structure. Okay. Okay, now the next question is, protein folding is a spontaneous process because, so why can you tell that protein folding is a spontaneous process? The del G is positive, the del S and del H are both positive, del S is negative and del H is positive, and del S of water is positive and del H is negative. So which of the following parameter is correct for the spontaneity of the protein folding process? So which parameter actually is important for determining the spontaneity of a reaction? So to understand the spontaneity of a reaction, it is the delta G, or the Gibbs free energy, that actually tells you about whether the process is going to be spontaneous, feasible or not. Okay, so but the delta G has to be negative. So in the first place, delta G is positive, which is incorrect. Delta S and delta H are both positive. So if your delta S is positive and your delta H is also positive, they can cut off. And so no information about the protein folding. You never know that delta H has to be, they cannot be both positive at the same time. Now if you look at delta S being positive, that is true, and your delta H has to be negative. So in the C case, it is delta S is negative, that is wrong. Then the entire T delta S term becomes positive and delta G shifts towards the positive term, making it non-spontaneous. Now delta S and delta H are both positive, that can be true. Now depends on whether delta H is more positive than delta S or less positive than delta S. So any difference between the two would actually matter whether it's going to be spontaneous or not. Rather, if you see that delta S is positive and delta H is negative, that will actually help the equation of delta G equals to delta H minus T delta S to be negative, and the process will become spontaneous. At the same time, if you see that what happens in the protein folding pathway is that there is a hydrophobic collapse of the polypeptide chain, that is leading to the increase in the entropy of the water, and an increased entropy of the water makes the value gives the value a positive term. Okay, and then what happens is that there is a change in the enthalpy, and that is very large and negative because of the formation of your hydrogen bonds, and you say the electrostatic interactions, van der Waals, and the interactions which contribute to this enthalpy term. And protein folding is, therefore we can tell that protein folding is driven by enthalpy and not any other parameter. So enthalpy is the major parameter which plays a very vital role in the protein folding pathway. So for question number 9, D is the right option. Okay, question number 10. Which of the following amino acid sequence is most likely to be part of the core of the protein? So just a hint for this question, the core of the protein consists of hydrophobic residues. So mostly the core of the protein has a higher concentration of hydrophobic residues. So now the work job of you people is to find out which has the maximum number of hydrophobic residues. Hydrophobic residues are the ones which have polar side chains or the side polar side groups. So try to recollect what was taught to you in the previous two lectures. So there we spoke about what are the charged residues, what are the polar group residues, polar side chain residues, and tell me which is the right option. Okay, so if you look back into the structure of the proteins, the amino acids, then you can, okay, so let us see what do we have. So then you can find out which has the maximum number of hydrophobic residues. So here we have lysine, proline, methionine, cysteine, and glycine. So proline, methionine, and glycine are hydrophobic in nature. So there is 3. How many do we have here? Phenylalanine, histidine, methionine, cysteine, and valine. So here we have 1, 2. Apart from cysteine, all others are there. So 4. Then we have lysine, proline, serine, threonine, and valine. Here also we have 1, 2. So here we have 2. And for the last one is arginine, histidine, methionine. So here we have 4. Okay, so histidine, methionine, glycine, and valine. So it can be both B as well as D. Now, in some cases, you do not consider the phenylalanine to be a hydrophobic residue. For in that case, you can give. Okay, so in that case, you can provide D as the right answer. So if you do not consider, in many cases, we do not consider phenylalanine to be hydrophobic. So in that case, you can consider D to be the right option. Okay, now the next question is, the homology modeling algorithm is applicable for. So what is the homology modeling algorithm actually conveying? So tell me. The proteins belonging to same family, belonging to different family, similar amino acid sequence, or having secondary structures with different linkers. Which is the right answer? Yes, that is correct. So proteins have to be of the same family, right? They cannot be of the different family. So 2 is anyway wrong. So if 2 is wrong, which options gets eliminated? Okay, this one gets eliminated. Okay, only one. Okay, let us see what is the with similar amino acid sequence. That is right. So 1 and 3. So 1 and 3. So this is also wrong because 3 is correct. So see, is 4 correct? Proteins having same secondary structures with different linkers? No, that is not correct. So the right answer is 1 and 3. Okay, very good. So proteins have to belong to the same family and have similar amino acid sequence. Good. The next question. What type of reaction occurs when a disulfide bond forms? What type of reaction is occurring when a disulfide bond is formed? These are basic level questions. These are questions which you can definitely get in your exams and your assignments. So these are easy questions. Quick answers for these. Very good. So it's the oxidation reaction. And this occurrence and breaking of disulfide bond is the redox reaction of reduction and oxidation. So you call it as the redox reaction. But when a disulfide bond is forming, it is the oxidation reaction. Very good. Which class of proteins is the heat shock protein part of? This is also an easy question. Heat shock proteins are part of which protein? Which class of proteins is heat shock protein? Catalytic proteins, chaperone-like proteins, immunoglobulins, ribonucleotide proteins. That is good. Chaperones. So HSPs are part of the chaperones. Correct. Okay. So this one. The structure of the protein. So is the example of dash structure. So what is the structure of this protein? Is it secondary structure, tertiary structure, quaternary structure, or primary structure? So you have a lot of alpha helices, you have turns, loops. You do not have beta strands though. Yes. So what is the structure telling you about? It's a secondary structure. Would you like to think a bit more? The protein seems to be well folded. If you have seen different structures of proteins, you would be understanding that this protein is a very nicely folded protein. There are a lot of secondary structures, but if you see that this protein is well folded in a very three-dimensional arrangement. Yes, so it's a tertiary structure. So let me tell you once again, the primary structure of a protein is its polypeptide chain. So the secondary structure is the regular recurring arrangement. So you have an alpha helix, you have a beta sheet. So these are separated. So they are present separately. So that is the secondary structure. Now the tertiary structure is a three-dimensional arrangement of the polypeptide chain as a whole. So you have to consider the protein as a whole, how the protein looks like. So that will actually tell you how the structure is. So it is a tertiary protein structure. Okay, never mind. Okay, you gave the right answer later on. B, that is correct. So B is the right answer. And the last question for the day, in the hydrophobic core of a protein, which of the following forces is responsible for the hydrophobic collapse? So what forces actually lead to the hydrophobic collapse in the hydrophobic core of a protein? Any other answer apart from D? Yes, so the non-polar surfaces, you see that for the non-polar surfaces of the amino acids helps in the building of the hydrophobic core. So due to the hydrophobic interaction, these um, so due to this van der Waals interaction, these hydrophobic residues can be involved in the forming the core of the particular protein and increasing the stability of the protein. So on breaking, if you need to break the hydrophobic core or break apart the hydrophobic core, you have to disrupt the van der Waals interaction. So these van der Waals interaction are very important for the formation of the hydrophobic core, and the loss of the hydrophobic core is because of the breakage of the van der Waals interactions or the van der Waals forces. So that's all for the today's class. Now if you have questions, you can ask me. So that's all from my side. Now if you have any questions, you can ask me.
So how can we reach you in case of any doubts, sir?
Okay, right. So I'm giving you my email ID. You can mail me. You can mail me in this.
Thank you, sir. Sir, will this be questions be uploaded, sir?
Yes, I would. This video is means this class is also being recorded. It will be uploaded in YouTube, and I would be sharing the slides with the NPTEL team, and they would be sharing it with you.
Yeah, thank you very much.
Any other questions? Okay. If there are no more questions, then I think we can end the class. Thank you all. We'll meet you next week.